All right, so for today's revision, we start with 2.2. So mainly we'll do chapter 2 today, as I said already. But if you're wondering where is 2.1, why did I skip 2.1? Because 2.1 actually nothing much. 2.1 is just mainly talking about the structure of the leaf, which honestly, nothing much. So we start with the main thing, the main processes, which actually starts from 2.2. So by the way, this chapter, if you're wondering what is it about, it's about the leaf, the Daun, right? Leaf. So inside leaves, there will be 3 processes we're going to learn about what happens in the leaf. So number 1 will be something called gaseous exchange, which is a very simple process. Okay, so before we start with gaseous exchange process, right? So remember, you need to know leaf, you have the upper surface and the lower surface. For gaseous exchange, it will typically occur at the lower part. So imagine this is the lower part of the leaf. I know upper, lower looks quite similar. So let's say now this is the lower part of the leaf. On the lower part of the leaf, right, there's a lot of holes, what we call as stomata. These holes are whereby gases go in and out. Oxygen come out, carbon dioxide come in, gases exchange. However, these holes, they are not 24/7 open one. Just like let's say if you run a business, you run a shop, your door also 24/7 open one, right? Usually open during when? During the day. Nighttime is when you close it. So same here. This stomata opens during the day and closes during the night. So there's actually something that controls the opening of a stomata. Just like your shop, you got security guy help you to open and close the door. Same here. There's like a security controlling the stomata called the guard cell, like literally called the guard, guards the stomata. So mainly here we're going to learn how this guard cell opens and closes the stomata. That's a process we must know how to explain. But before we start with the process, right, you need to know one basic thing. Wait, let me copy it from here first. Yeah, never mind. Okay, we still restart here, draw it here. So you need to know one basic thing. So for my students, this one actually you're quite pro already, like we mentioned this so many times already, which is you need to know something called hypo and hypertonic solution. So a very basic concept in bio, you need to know this. So hypertonic is a solution that has more water, lesser solute. So solute basically is a substance that you put in water to let it dissolve, like sugar or most commonly even salt. Hypertonic is the opposite. This solution has lesser water and more solute. So when you have, let's say if you have salt water, if you put a lot of water, right, you won't taste so much of the salt, right? Your solution will be more like diluted. So imagine, let's say you have a cup, you have salt inside. Okay, and then if you put a lot of water, it will be very diluted, right? You don't taste the salt a lot. That's why a lot of water means low concentration of solute. Similar to hyper, if let's say you put very little water, your water will be more salty, it will be more concentrated. You can taste the salt more. Yeah, that's why lesser water will give you more solute. So you just remember hypotonic is like the animal hippo, more water. All right. Yeah. So if you put these 2 solutions side by side, remember water will always flow from more to less, high to low water concentration. So water more to less, which means water will always flow from hypo to hyper. Get it? Yeah. So this is a very simple concept in bio you need to know first before we can start this. In case some of you still don't know what's this. So yeah, here's the mechanism of opening and closing of stomata. So this is the guard cell that I was saying, that this is the penjaga pintu. This further is the one that closes and opens the stomata. So you must know the label. So this, if they ask you what is this, you must say this is the guard cell. Look at the hole in between the guard cell. Yeah, that is the stomata actually. Sometimes we do refer it as stoma, but we follow our textbook, we say stomata. Cannot? Now, so as I said, daytime is where the guard cell opens it and nighttime it closes it. So now you must know how to explain the process, why daytime the guard cell will open it and why nighttime it will closes it. So to remember the process easily, right? Here I will write down a few key steps for you to help you remember. Okay, first, photosynthesis. So when it's daytime, definitely photosynthesis occurs. So gut cell actually has a lot of chloroplasts. Okay, that's why in gut cell, what do you call that? Photosynthesis actually occurs. Okay, so there will be photosynthesis during daytime. So you should know photosynthesis in plant is to mainly produce what? Glucose. When there's glucose, there's sugar, there will be energy. All living things require glucose to produce energy. So we mentioned this first. Come. So we write here together. Come. Here we are, write. If you don't know notes, also need to write. So photosynthesis occurs in gut cell. So therefore, more glucose, you say, is produced to generate more energy. And what is this energy for? You will write this one real quick, then we go to the next point. So the next one here, energy is for a process called active transport. So active transport also is in the same chapter as this hyper. So active transport is a transport that requires energy. That's why it's called active transport. Okay, so when there's energy, then this active transport can actually occur. So you can say for active transport. So point number 2, basically, they're all linked together. One got glucose, got energy, got active transport. So what I want to transport actively right now, what is this active transport going to transport? I'm going to transport something called potassium ions. All right. So now inside your gut cell, because there's photosynthesis occurring here, there's a lot of energy inside here. So actually what's around your gut cells, right, are basically the leaf cells. Okay, because if you look back at here, right, see these are the stomata with the gut cells, right? Around the leaf, right, there's also a lot of other cells around here on the surface of the leaf. So all these cells, we're going to call it as adjacent cells, means the cells surrounding the stomata. So since there's a lot of energy here, it will cause potassium ions from the surrounding cells to be transported into the gut cell using this active transport. So now I got energy, I can use energy to transport them into the cell. Understand? So potassium ions, potassium actually is a kind of salt. So right now, there's like a lot of salt inside my gut cell already. So there's a lot of salt in your gut cell now becomes hypertonic because there's a lot of salt as compared to the outside cell. So the outside cell loses the salt to the gut cell, right? And so there's not much salt left, so it becomes hypo. So basically now outside here is hypotonic. Inside here becomes hyper. Okay, let's explain this first. So we are until here right now. So when there's energy, there's active transport, potassium ions diffuses into, you can say diffuses from the surrounding cells, right? Called adjacent cells into gut cells. Yeah. And once the ions are into the gut cell, they like have a lot of salt already, right? If you eat a lot of salt, you'll be hypertonic. So the gut cell becomes hypertonic. Towards the adjacent cell. So hypo, you always have to compare between two regions. Right? So if the gut cell is hyper, the outside region, which is the adjacent cell, will be hypo. So we say hyper towards the adjacent cells. Right? So right now, there's a lot of salt inside your gut cell. it's hypertonic compared to the outside. So remember I told you water will always flow from hypo to hyper, more water to less water. So this is going to cause the water from outside to be flushed into the gut cell. So the process of water flowing from one region to another is actually called osmosis. I believe most of you already know this, even though you're not my student, right? So osmosis means flow of water. So water will now flow from hypo to hyper outside the cell into the cell. Okay, just remember when you eat salt, you'll be very thirsty, right? So if you have a lot of salt, you'll be hyper, you need to drink water. So it's the same thing here. When you have a lot of salt, you need water. So water will come in now. Okay. So right now, not just the salt going to come into the gut cell, water also going into the gut cell. When water going into the gut cell, right, you can see the gut cell expand. And this is why the gut cell can open the stomata, because this is the original shape of the gut cell. When there's water going into it, it will become from this to like this. So it basically expand and then open up the stomata. And that's how the gut cell opens the stomata during the day. So now we just have to say the water goes in. So water then diffuses into gut cell by osmosis and becomes, so we can use this word, turgid. So turgid is a term used to describe a plant cell expand already, which is what you can see in the diagram here. Okay, understand? Cannot? So yep, osmosis done. So you just need to use these key points to help you explain the opening and closing of the stomata. Cannot? Right. So if can already, now later you try to write the nighttime one by yourself, then we check our answers later. But before that, let me briefly go through first. So how do we now explain this one? So yeah, questions here could come up for essay. Like they might ask you to explain how the stomata opens during the day or how does it closes during the night. So this is a very straightforward process, very straightforward kind of question if they ask like that. So nighttime basically is the total opposite. So why the stomata close? What causes it to close? Same thing, we start from photosynthesis. As we know, nighttime, no sunlight, no photosynthesis. No photosynthesis means there won't be glucose, there won't be energy generated for the so-called active transport just now. So the first sentence I give you, okay, no glucose is produced and no energy is generated for active transport. So right now, the potassium ions they have gathered inside the gut cell during the day, right, they will stop coming inside from the outside already. So what happens now is the potassium ions will then diffuse from high to low concentration, because when I go from high to low, I don't need energy. So it will come out very easily. Right? So right now, all these come out already. So if all the potassium ions come out, there's no more salt. So you can see lesser salt means it will be hypotonic. So right now, if you compare the adjacent cell and the gut cell, your gut cell now becomes the opposite now, hypo, and outside is now hyper because outside has the salt right now. So water will always flow from more to less, hypo to hyper. So water is going to come out from the gut cell. And once that's happened, it will go back to its original shape. It will shrink. And then as you can see, it closes the stomata. So when the gut cell shrink, right, there are two terms you can use to describe it. You either can say the gut cell becomes flaccid, or you can use the word it undergoes plasmolysis. Okay, so I give you guys around 1.5 minutes. The 2 minutes a bit too long, like that, right? Yeah. So 1.5 minutes, you'll try to write this part yourself. So if there's no active transport, then what happened to my potassium ions? So I already told you everything. Okay, you'll try to write this. Then later we check our answers. Okay, come on, go, go, go. Don't lazy, Come. If you lazy, then you will gonna curse SPM will fail. Faster, come. Later we'll cross check our answer. So far done. Okay, come. So one by one, I review each sentence. So just now the potassium ions diffuse in, right? So now, of course, it goes the opposite direction. It diffuses out. So once it goes out, the sort change from one side go to another side. So the hypo also change. So next thing we talk about the hypo. So now the gut cell instead of hyper now becomes hypotonic. There's more water inside already now. So the water will come out. Okay, water diffuses out of gut cell by the same process, osmosis, and it becomes flaccid, or you can say undergoes osmosis. So right now, basically, it curves back inside, go back to its original shape, closes the stomata. And that's how we explain this. Okay, so this is considered very straightforward in your question. So once you're done with this, if you don't have problem, okay, let's look at the second situation here. Now, this is something they like to ask quite often also, which is they say, why stomata sometimes are found to be closed on a hot day? So for my students, right, this one we've done before already, right? Remember or not, guys? Even though it's daytime, right now just slightly hotter, the stomata close. It's daytime, or remember, we still got sunlight. Or even though we mentioned all this already, got photosynthesis and all, but they still close sometimes. How? Why? So you need to explain. Remember or not? Why it closes? Come on, guys. Remember or not? Okay, Renia, that is actually the explanation, but you got to talk about the process that causes it to close. Remember or not? Divya, remember? Jingshen, do you remember? Angeline, remember or not? So a lot of you say to prevent excess loss of water. Some of you prevent loss of water is actually the explanation. Yes, I know I want to close it during hot day because I don't let water come out from the leaf. But how the stomata, like the gut cell stomata know, right? Hey, hot or I must close. So you got to explain that part. Okay, Georgina, very good. Exactly correct. Zingshen, okay, correct. Water evaporates from your stomata. I mean, not from your stomata, from your gut cell. Yeah. So excess water evaporates from gut cell because remember, gut cell is on the surface of your leaf. So if the surrounding is very hot, water will still evaporate to the surrounding. Okay, let me erase all of this first, a bit messy here. Yeah, so water basically will evaporate to the surrounding. Now, even though daytime, right, you have osmosis happening, water going into the gut cell, but the water evaporates faster when it's too hot compared to the water coming in. So overall, I will lose water. So basically, your gut cell from this becomes this, even though during the day, because I have too much water evaporate from gut cell. So therefore, we will say, just like the nighttime, gut cell becomes flaccid and stomata closes. Then only we state why they want to do that. What's the purpose of this? Like most of you mentioned just now, it is to prevent excessive loss of water. So a lot of you will seriously say, oh, I want to close during hot day because I want to prevent excessive loss of water. But usually this will be a 3-mark question. You will need to explain like how you close. Then only you say why I want to close. Understand? Cannot? Yeah, but if it's a 1-mark question, then yeah, you can straight away mention how to prevent excess loss of water. But this question needs you to explain. So yeah, you need to write this off. Right? So once you've done 2.2, that's it, finish, settle. Usually here not much questions. The questions they ask also is quite straightforward like that one. Yeah, so let's try. I'm going to try, but let's straight away go to page 11. Now we go to the next topic already, transpiration. One, two, three. Okay, done. Let's go. Now, Main organ for transpiration. So by the way, transpiration is the process of, as you can see, water evaporate from the leaf. And how water evaporates from the leaf, it also occurs through the same hole we mentioned just now, stomata. Yeah, so stomata can basically have two functions, gaseous exchange and for transpiration. So besides gases go in and out, water also come out from the same exact lobang. Yeah, but here, even though they show you it's like on the top of the leaf, but it usually occurs on the bottom, because like we mentioned, stomata usually on the bottom of the leaf, right? Okay, so yeah, transpiration. It might sound, every time we talk about transpiration, right, you all think, oh, loss of water through leaf, it's a bad thing for plants. But sometimes plants, they undergo transpiration on purpose because there's actually benefits towards the plant. For example, look at here, here's the importance. Number one is say it helps in absorption and transfer of water, mineral ions from roots to all parts of the plant. So yeah, for my students, recently we learned this in chapter 4, right? In plants, right, water is like a continuous chain from the leaf all the way to the stem, to the roots in the soil. Water is like a chain interconnected with each other. So once transpiration occurs on the surface of the leaf, right, it will basically pull this entire chain of water up. So hence helping the plant to intake water and salts from the soil. So this is actually a good thing. So when this happens, right, it can always make sure there's water going into the plant. Unless, of course, the soil dries up, then that's another story. And number two, right, transpiration, you can imagine just like sweating, like you can see water coming out from the leaf, it's like water coming out from the skin. Why do we sweat? We sweat when we basically feel hot, right? Because this can produce a cooling effect. You can remove excess heat, eliminate heat. And just like human also, sweating also helps us to remove excess water. When you drink too much water, you need to go toilet pee, you remove the excess water, and that's how they do it. Because sometimes when it rains, they might intake too much water. They need to also find ways to get rid of it. So this is how they get rid of it, through transpiration. Right? So in transpiration, most of the question, right, will be very closely linked to this part, the factors. So factors of transpiration is the main thing you need to focus in 2.3. So there are 4 things that can affect how fast or how slow transpiration can occur. Now, to easily remember the factor, right, now you don't think about transpiration, you think about water evaporating, water drying, because transpiration basically is water evaporate, like the water dry up on the floor. So let's say if you pour water on the floor, or you mandi your hair very wet. So what can cause the water on the floor or the water on your hair to dry up faster? So let's say you have your floor, your floor is here. The floor very wet, you pour water. So besides mopping the floor, what could cause the water to evaporate fast? Number one is temperature. Right? If it's hot, the water evaporates faster. Just like you put your baju under the sun, it dries up faster. Right? So a bit common sense. And number 2, again, we imagine your hair very wet after you mandi already. Okay, so hair wet, what do? You take the hair dryer and you blow your hair, right? Because when got wind blow, the water will be blown away from your hair. So air movement is another factor. All right, and then light intensity. So just like, why do we hang our clothes when there's sun? Because when there's light, usually there'll be heat along with it that cause your water to dry up faster, right? Yeah, so just remember sun. And then lastly is air humidity. Humid, you all know what is that, kelembapan. So imagine right now, if you put a towel over your wet hair, cover your hair. Imagine you mandi your hair very wet, you cover with a towel. So if you cover your hair with towel, right, do you think your hair can dry easily or not? Of course not, right? Because if you cover your wet hair with towel, it will trap the moisture, trap the water inside your hair. So it'll be very humid inside. So cannot dry, right? So that's how humidity affects evaporation, transpiration. So basically, when it's very humid, the water dries very slow. But if you open your hair, the water exposes more to surroundings, so it'll be less humid, it dries up faster. All right? So let's go through one by one. So light intensity, now, even though I say more light, more heat, right? But for the explanation, you cannot explain like that because, yeah, more light got more transpiration because it's related to what we mentioned just now. When there's more sunlight, there will be more photosynthesis in gut cell. And when there's photosynthesis in gut cell, your stomata open. And when stomata open, then water evaporate. Yeah, so when you explain light intensity, you've got to use what we mentioned just now to explain. Temperature will be more straightforward. So hot, then dry faster. So higher temperature, higher rate of transpiration because it increases the temperature, increase in temperature, increase the rate of evaporation. It's that simple. Then air movement, remember, is just basically wind. Just remember hair dryer. You blow your hair, it dries out faster, right? So more air movement, more transpiration, more water will be carried away on the surface of lid. And lastly is humidity. Now, humidity, you can remember like this. When the air is humid, like you cover your head with a towel, you trap water inside your hair there. So if outside is very humid, means the air, there's a lot of water. So this is actually linked to osmosis, like we mentioned just now. Water always flows from more to less. If outside got a lot of water already, water will not flow out from the leaf anymore, right? Because water is high to low, not go towards high. But let's say if the air is very dry, there's less humid. Now the water potential is lower outside. So this is just like osmosis. The water can go out easily. Get it? So they say, so humidity, by the way, is the only factor whereby it's inversely proportional. Higher the humidity, lower the rate of transpiration. Okay, because your atmosphere have higher water potential, which reduce evaporation. That's simple. Okay. Right. So that's it. But for the factors, right, question won't ask like this direct. They won't say, oh, explain how light intensity affect transpiration. Usually they won't ask so direct. Usually they will try to link certain situation to the factors. For example, they give you a certain diagram or certain scenario, then you got to think, oh, what factor does this scenario has which affect the transpiration? So for example, okay, here I give you a few examples. Explain how does each of the following conditions affect the rate of transpiration in plants. So usually this is how I carry out revision. Okay, you will write more than usual class. So at the same time, you will create your short notes. When you write, it's like you're answering question like that. So you remember better. It's like doing question. And I'll put like certain short questions together with it. So your notes got questions at the same time with it. So yeah, but no, I know you don't have notes now, but just write down first lah. Okay, so for transpiration, you need to know this experiment. We'll come up quite often one under transpiration. So this experiment, you require this apparatus called photometer. Yeah, this whole thing here is called photometer. And then you can see that this photometer comes with a capillary tube. So this part is called the capillary tube. The entire setup is called photometer. Specifically, this part is called capillary tube. Now, for those who don't understand this experiment, okay, let me explain. Now, basically what happened here is I put a plant at one end of the photometer, and I make sure here is airtight. There's a cork here covering the air spaces and all, and you'll be soaked into water. So you can see inside the capillary tube is basically filled completely with water, except for there's one small air bubble here. I purposely allow an air bubble to enter here. So what's going to happen here is like this. You can see here, I set 2 points, M and N. All right. Now, when transpiration occurs, this air bubble is going to move towards the plant. Okay. More transpiration means you have more movement. Right? Why? It's because we mentioned just now already. When transpiration occurs, water is like a chain interconnected with each other from the leaf all the way to the roots. So when transpiration occurs, it basically sucks water up into the plant, which causes the air bubble to move towards the plant. So usually what they do here is like this. They will set 2 points, let's say maybe on 5 cm or 10 cm. Then they will record the time taken for this air bubble to move from M to N. So technically, if the transpiration is high, the time taken will be much more faster. This is not slower. This is lower time taken, means shorter time. Shorter means faster. Understand? Because you suck water faster, so this thing will move faster. But sometimes you need to take note. Sometimes the experiment, they will change a bit. Instead of setting 2 points, they don't have these 2 points. All they have here is a ruler. So instead of fixing the distance, they will fix the time. So like they will set the time 10 minutes and do a countdown. At the end of 10 minutes, they will measure the distance traveled by the air bubble. So of course, the more you move within these 10 minutes, the higher the transpiration. The lower you move, the slower. Yeah, so it depends how they carry out. There are a few different methods. You need to pay attention to the question. So for this one, let's redo the first one here. They say, explain the time taken for air bubble to move from point M to N as the lamp is moved closer towards the plant should. Now, I want you guys to first tell me the time taken. If I move the lamp towards the plant, do you think the time will be faster or slower? Let's answer the first part first. So you got to think, when I move the lamp closer, right, what is the factor here that affect? Does the transpiration increase or decrease? And hence the time, will it increase, be faster or slower? Okay, I want you to tell me the time first. Fast or slow? Fast, 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 fast. Yeah, the factor is intensity of light. But first, you got to mention this, right? Time taken will be how? Answer is fast, right? Like most of you say here, fast. Those who didn't say fast one, slow is wrong. It'll be faster because if I move the light closer, more light intensity, right? More light, more transpiration. So it will be fast. It will move faster. So let's start with that first. So a lot of you, right, when you do this kind of explanation question, right, you all will left out this part and fast, explain, you all faster, right? Explain. Yeah, and then you all forgot to answer the main question. So if you don't answer the main question and straight away write the explanation, you won't get the full 3 marks. Yeah, so sometimes you all might know the reason, you all know the answer, but then it comes down to carelessness instead in terms of understanding the question. So at the same time, also teach you how to fully understand, read the question. So you must start with time taken. So you can say the time taken will be slower, shorter, or you can say faster. All right, why faster? Then not only we talk about the factors, transpiration, or so you can say because when you move the light closer, there'll be more light intensity, right? Because light intensity increases as the lamp is moved closer to the plant should. Then more light, more transpiration. So you just say rate of transpiration increase. Just nice 3 points. Okay, I give you one more bonus point. Whenever you do this photometer experiment, right, there's one more explanation we can include, which is not mentioned here. Because since you're doing photometer experiment, why more transpiration that this thing will move faster? Because you absorb more water. So that's an additional point we can say. So let's say in 4 marks, then you got the 4th point here. You can say, hence more water is absorbed by the plant should. That's why the thing moves faster. Okay, let's try another one. Example 2. Explain the condition of the leaf when the fan is turned on. So yeah, focus on this part first. Condition of the leaves. So after when I turn on the fan, let's say around like a few minutes later or a few hours later, what can you observe on your leaf? Let's talk about the leaf first. What do you think will happen to this leaf? Will it fly into the air or become even more pretty? That's what it means by condition. What happened to it? So, dry. Now, I prefer not to use the word dry. I prefer to use another more specific term. So when leaf dry, what do we call it? Okay, Nadini, correct. That's the exact term. So we will say the leaf or what happened. When leaf dry, we will say what the leaf will wilt. So that is the term we need to mention. So that is the condition. Okay, so you can say the leaves will become wilted. Or let's say after 1 hour because you turn on immediately, it will wilt, so it needs time. So now only we explain also same. To explain this, we use the factors that we mentioned, that we learned just now to explain. Also, what factor is this? You on the fan is wind. Wind is air movement law. So we say air movement increases when the fan is turned on. Therefore, we say here, rate of transpiration increases. And I give you one more point here. You also can say, rate of transpiration increase because more water carried away from the surface of the leaf. You also can say that. Additional point, more water is carried away from surface. Let's look at the third one. Okay, the third one, see the question, I didn't put it construct very well. Okay, but never mind, I will let you know what it is. So they say here one plant. Oh, sorry. Too fast. Right, let's go. So next one here is they say here one plant is covered with plastic bag while the other is not. Compare the condition of plant after 2 days. Now the condition here, actually both plants, they won't die of course, because assuming like let's say water, everything is given to them, they have everything, food and all, okay. So now what will happen here is, right, okay, I want you to tell me that which plant will be more turgid after 2 days. Means which plant will actually look like slightly more healthier, will retain more water in it. 1 or 2. So one covered with plastic, one didn't cover. So which one will look more turgid, like look healthier a bit, look like a more water in it? Got 2, 1, 1, 1, 2, 1, 2, 2, 1. Okay, quite divided also. Now the answer actually is it's 2. Why 2? Remember, this is just like putting the towel on your wet hair. If you cover your hair, wet hair with towel, can the water from your hair dry up? No, right? Same thing here. If you cover your plant with this plastic bag, water is not going to evaporate a lot from your leaves. Of course, you need to leave a few gaps for the plant now. Okay, if not, they have no gas. So imagine assuming there's a few holes given to them. if I cover them, the factor here we need to explain is actually humidity. So the plant have higher humidity. So we mentioned now higher humidity, lower transpiration means I will retain more water in it. So the answer actually is 2. Okay, so this question actually we got do in class before. When you cover plant is low transpiration. Low transpiration means you're going to retain more water. It's better. So we can say plant with plastic bag will be more turgid. The reason is because air humidity is higher. So therefore, rate of transpiration is lower. Ta-da! All right. So for transpiration, actually, I only got prepared one question for transpiration, which is page 19. The question book so question and notes actually separate into 2 sets. So for the question one is on page 19. Yeah, you can try to do this yourself. So you can see the experiment go on photometer. So the apparatus shown here is photometer. So for this one is they give you a specific distance and you need sorry, not specific distance. They give you a specific time, not specific distance, and you need to record the distance traveled by the air bubble within that specific time. Okay, yeah, so slightly different this experiment, but the concept is the same means more transpiration means it will move more. So I want to do a bit definition punya question Okay, let's write this together over here. What is meant by transpiration? So transpiration. Okay, let me ask you guys. Your English don't have to be too good to answer this question. So just now when we talk about transpiration, right? What is the keyword do you think I need to mention in this answer? Like when we talk about transpiration in plants, in leaf, right? Means it involves what happening? Okay, Nandini, very good. Exactly what I want. So others, what do you guys think? Transpiration in plant involve what happening? Involve what happening? Xinhui, exactly correct. Georgina, almost there. Yeah, I prefer another word compared to loss of water. Quite a lot of you say loss of water, but it would be better to say when you lose water, how they lose water? They lose water by evaporation. So they are hole open. They don't pour the water out, right? The water just go out by itself. So that's called evaporate. It's like I open my bottle cap and let it sit in the living room. So the water lost through evaporation. So that's the key word, evaporation. Okay, so we will say transpiration is the evaporation of water. Okay, you can say in the form of water vapor. And through where? You can say that through the surface of plant. Or you can say through stomata also can of plant. Okay, yeah, so the rest is quite easy one lah. You try to explain yourself. Oh, for this one, okay, I just want to write for you also lah. How does the rate of transpiration is measured in using the apparatus above? So the how do I measure the transpiration here? Remember, is you measure the distance traveled. Yeah, so it's not a bit technical, a bit we say by measuring the distance traveled by air bubble in a given time. Let's say 10 minutes. All right. So once you've done this, There are others you try to do yourself. Like I say, answers is really given in the link that I post just now in the chat there. And also, when you receive the notes, there will be an answer inside it also lah. So let's now go to the most important topic of chapter 2, which is the photosynthesis. Okay, but before we go to photosynthesis, okay, I give you a 2-minute toilet break. Okay, since class is 2 hours instead of usual 1 hour. Okay, you'll take 2 minutes. Okay, go wash your face, toilet, then we come back, photosynthesis, then done. Okay, come. See you in 2 minutes. So we're already done with gaseous exchange, we're done with transpiration. So this is the final process in living organism, photosynthesis, which is also the longest one. Yeah, it's a bit tough lah. Yeah, but photosynthesis is actually a chemical reaction. So actually it's quite involved chemistry, because this is a chemical reaction, there'll be a chemical equation involved. So photosynthesis is a chemical reaction that occurs in living things. Hence, we call it actually a biochemical reaction. So by now, if you still don't know the difference between biochemical and chemical reaction, chemical reaction is a reaction that occurs outside of living things. So if it occurs inside your body, inside plants, you are a living thing, so you add the word bio in front of. So you can see, that's why we have a chemical equation over here, which represent the whole photosynthesis process now. So in photosynthesis, you mainly need these two materials, water and carbon dioxide. And also you need sunlight. Now, sunlight is not really considered a material, because material means something that is non-renewable, but this is renewable. But anyways, you still need sunlight, so we include it as our material as well. And you use these three things to produce oxygen and mainly glucose. So glucose is the main product that plants want to produce through photosynthesis. So that is their so-called food that we always say. Okay, but if you look at your textbook, right, this equation actually got water behind. Now, if you look up in Google, right, I think I told you all this before, there won't be water in this equation. However, we just follow our textbook. Now, why is there water behind? It's because they use more water in front, and then somehow they release a bit behind. So it's just like you drink 1 liter of water, but you pee out 200 ml. So they will generally take in excess water, but they will produce a bit behind. Yeah, but yeah, so you just include the water here when we write the equation. Now I'll explain why is the water here later. So before we get into the photosynthesis process, you need to know how the leaf carries out photosynthesis. Okay, you must know how the structures in the leaf involved in photosynthesis. So usually you'll see questions asking like this: how the leaf structure is adapted for photosynthesis. Now, before we answer this question, you must know how to label the parts first. You must know what parts are inside the leaf. Then only you know what part, how each part are adapted for photosynthesis. So we start from the topmost layer, which is called the cuticle. So this cuticle is that smooth surface on the leaf that you touch on. So leaf, we mentioned before, right, there's a smooth and a rough surface. So the top is always smooth. So that smooth surface is actually called cuticle. Right? Then below the cuticle is like the skin of the leaf. This is called the upper epidermis. So if this is called the upper epidermis, the upper skin, then this lower skin is basically just the opposite, called the lower epidermis. Now inside the leaf, these two parts are the main structure of the leaf, which not just supports the leaf, but also mainly carry out photosynthesis. So the first one, it's called Pelicip Mesophyll. I remember PMS, Pelicip Mesophyll cell. And then the second one, you can see the shape. It looks like the sponge that you wash your plate at home. That's why it's called Spongy Mesophyll cell. And then look at this round thing. So this round thing is actually the veins that you can see on the surface of the leaf. All these veins you can see. Yeah, so this thing is basically called the vascular bundle. Okay, so for my students, we always mention this vascular. For the past few lessons, right, we've talked a lot about vascular. So whenever we see vascular, means inside got what? Vascular got what and what? It's always these two things only, which is correct, the xylem and the phloem. Yes, so that's what vascular is. Inside these veins is actually got xylem transporting water and phloem transporting the glucose away from the leaf. Yeah, so it's basically just xylem and phloem. Yeah, easy, this one. Yeah, then we move to the bottom of the leaf. So at the lower epidermis is where you can find, just now we mentioned those two law. This is basically the guard cell. And yeah, this is the hole, the stomata. Oops. Now actually our bottom of leaf don't have cuticle one, even though they put cuticle over here, because most leaf only have cuticle on the top. So only certain leaf have cuticle on the bottom, but generally they don't have at the bottom. So we will learn it as no cuticle at the bottom, only on the top. Must we label the pericycle or don't need? No, don't need to be too specific. So what are we labeling here are basically the tissues and the cells of the leaf. So remember, leaf is actually an organ. So organ are made out of tissues. So this is a tissue, this is a tissue, this is a tissue. Yeah, just remember, we just label the whole tissue can already. All right. Now, for this question, how the leaf structure is adaptated for photosynthesis. So to get 10 marks, if this come in an essay, maximum is 10 marks. You can come out in structure, usually 3 or 4 marks. But if you come in an essay, right, how to get 10 marks? You actually only need to explain 3 things, then you get 10 marks already. So this one, right, we study smart. You don't memorize everything. You just need to memorize, not memorize, you just need to remember. This one, cuticle, are the one that I wrote in red color. Cuticle, Pelicip, and Spongy. So Pelicip and Spongy must mention because these two is the main part that carry out photosynthesis. Okay, so let's start with the cuticle first. So just now we mentioned cuticle is the smooth surface on the leaf, right? Now remember, why is it smooth or not? What makes the cuticle smooth? Because it's made out of what? It's a waxy layer. Yes, Radiant Xingxuan Nandini, very good. So it's a waxy layer, so, which has what characteristics? So in this case, waxy. What is the key characteristics of wax here? Yes, it is waterproof. Why does it have to be waterproof? Because I want to prevent water loss from a leaf. As you know, leaf is always directly exposed to sunlight, which causes water to evaporate easily. And that's the reason why we put our stomata at the bottom of a leaf. Because if I put it at the bottom, it won't be directly exposed to sunlight, hence reducing transpiration. Okay, so but on the top, I cover it with the cuticle. So you can say, like we mentioned, to prevent excessive loss of water in leaves. On top of that, there's one more thing we can talk about cuticle. So wax, if you look at wax, usually, of course, they are waterproof and they are also wax. Oh, Nanisha, you tell me already. Yeah, transparent. Yeah, correct. You can say transparent. So just now, the main thing I want to focus is waterproof. Yeah. So transparent, yep, is also you can say. So you can say cuticle is also transparent. No? Why? It's because you want to allow penetration of sunlight. No? So, one thing I forgot to add just now. For the adaptation, right, you're going to describe, when they ask you to describe how the adaptation of each structure, right, you got to link the adaptation towards these 3 things. Like what makes each structure are good in absorbing water, carbon dioxide, and sunlight. Because if you absorb these three, they only can photosynthesis. So actually, all the adaptation we're about to explain, right, is related to absorbing water, carbon dioxide, sunlight. So you can see here, we talk about water, we talk about sunlight. Let's move on to the next one, Pelicip Mesophyll. So for Pelicip Mesophyll and Spongy Mesophyll, right, the key thing you need to explain here is how are they arranged? Yeah, so their arrangement is very important towards how they can absorb either sunlight, water, carbon dioxide. Okay, so for Pelicip Mesophyll, they mainly absorb sunlight one. So right here, Pelicip Mesophyll cell. So remember what's their arrangement? How are they arranged? So their arrangement, number one, you can see they are very packed, correct? So they are very close to each other. So if you compare Pelicip and Spongy, right, they are so much closer to each other, right? As compared to here, there's a lot of gaps. Apart from maybe this one a bit chacha, like see here got a big gap. Okay, this fella should go closer. And besides that, they are also arranged, standing upright. So you can see all the cells is like standing, right, like straight. So that is the two things you could talk about. Are arranged upright, means they're standing, and packed tightly. And what's that for? Why do you want to arrange like that, leh? For what? How does it help them to absorb sunlight? It's because if you are packed tightly, there's no gaps in between you. So whatever sunlight that you are exposed to can directly go into you. So you can see this fella, see, if he stand far apart like this, got gap, right, the sunlight then can pass through him. So you waste sunlight. You don't 100% absorb all the sunlight you're exposed to. And if you are standing up, you basically can fit in more Pelicip. So if you can fit in more Pelicip, you can take in more sunlight. You can fit in more sunlight as a whole. Okay, because if you're lying down, you take up more space. Lying down is like you are long. Yeah, so they have to stand up. So you can put in more Pelicip and therefore absorb more sunlight. So you can say this is to absorb maximum. Yeah, so the keyword here must say maximum sunlight. And besides that, in order to fully absorb sunlight, Pelicip Mesophyll is the cell inside plants that have the highest amount of chloroplast. Chloroplast is the main organelle that absorbs sunlight. So you've got to say they also have high number of chloroplasts. Same thing lah, to absorb maximum sunlight. Done. So next, we're going to look at Spongy Mesophyll. So for Spongy Mesophyll, they are mainly involved in absorbing anything other than sunlight, which is the remaining two, water, carbon dioxide. So in order to absorb water and carbon dioxide, now same thing, you got to talk about their structure, their arrangement. So their arrangement, their structure, all is the complete opposite of Pelicip. So just now we start with packed tightly. So now it's packed tightly, right? So this one is actually, you can see they are further apart. So what's the opposite of tight? Tight is loose. Yes, correct, exactly. So we say they are arranged loosely. So that is the thing here. Spongy Mesophyll cells are arranged loosely. When they are loose, there's a lot of gaps in between them. So these gaps, like Nandini mentioned very good, are called air spaces. So we can say which forms air spaces. So this, when I have air spaces, like these gaps here, means, right, when carbon dioxide comes into here, they can easily go in between them and easily go into the cell. Same thing, when water comes from the xylem, it can easily go in between them and diffuse into the cell. Okay, so you can say forms air spaces to allow diffusion of gases and water. And number 9, their structure. So just most plant cells, right, they are rectangular shape, square shape. For this one, their shape is like an animal cell, like amoeba, like that. Yeah, we will say they have irregular shape. So you can say have also irregular shape. Now, how does irregular shape helps them in this case? So instead of like square, their shape is like amoeba, like that, curve in and out, have curves, you know. Curves is what makes you beautiful, right? So why are they curved like that? Yes, to give them a larger surface area. So if their surface area is larger, right, you can see what. See, usually if their surface like this, right, gas only have this surface to come inside, this amount of surface. But if they are like that, see, gas can go into here, here, here. There are more surface area. So imagine that your door is wider. You can go in and out from your house easier, right? So this is 2. Keyword surface area to increase surface area for these 2 for same thing of diffusion of gases and water. Ta-da! Just nice 10 marks. All right, so now we're going to move on to the main topic for today, which is photosynthesis punya process. So I realized a few people just joined us. Okay, never mind. You joined at the right time because this topic is the hardest of today. And also, yeah, most questions can come out from here. The harder question for this chapter will be from this part. So which is the photosynthesis punya process. So remember, photosynthesis occurs specifically in the chloroplast. So it's not like you say, oh, plants photosynthesis means everywhere can photosynthesis. No. So actually upper epidermis, lower epidermis cannot photosynthesis. Xylem cannot photosynthesis because they don't have chloroplast. So Pelicip, Spongy have chloroplast. And earlier today we mentioned the gut cell have chloroplast. So actually, yeah, Spongy also got chloroplast, just not as much as Pelicip. So inside the chloroplast, this is how it looks like. There are 2 main structures inside the chloroplast that you must know how to label, which is the granum and stroma. Because granum and stroma is whereby the 2 main reactions of photosynthesis occur. So photosynthesis is divided into these 2 reactions. You can see on my left here, we have light-dependent reaction, whereby from now on, I will call it as LDR, easier to read out, and light-independent reaction, LIR. So if you look inside your chloroplast, there's this stack of pancakes like that, right? So those are called granum. And granum, each individual layer, right, is actually called thylakoids over here. So if the question asks you to label this, if they point 1 digit, thylakoid. If they point the whole layer, that is called granum. Yeah, so usually this stack of pancakes will be connected like this. And then the other part, stroma. So stroma is just the empty part of the chloroplast. It's called stroma. Now, for granum, it's the part that contains chlorophyll. Yeah, by the way, chlorophyll and chloroplast are not the same. Chloroplast is the whole BG. Chlorophyll is a green pigment, a substance inside the chloroplast, which is typically found inside the pancakes inside the granum. Okay, so chlorophyll is basically the main thing that absorbs sunlight. That's why the reaction that requires sunlight, called light-dependent reaction, LDR, will actually occur here. Because they need sunlight, they depend on light. But for the other one, light independent LIR, it is independent from light. It does not require light. Hence, it does not need to occur there. It can occur here, the stroma, which doesn't have chlorophyll. So for LDR and LIR, they are actually both different types of chemical reaction. So hence, the easiest way to understand LDR and LIR, right, is by using chemical equation like chemistry. Okay, but the chemical equation here are very simple only one, don't worry. So yeah, if you look at LDR and LIR, both of them have a specific chemical process called photolysis of water and reduction of carbon dioxide. Yeah, see the word reduction. It's basically a chemistry thing. Yeah, so these are the two processes inside each of the reaction. Now, before we start going through the processes, right? Now, this question, the most basic way of question asking this part, right, is to explain the reaction in photosynthesis. So you need to explain LDR and LIR, total 10 marks for essay. So you can start your essay like this. Oh yeah, you have to start with LDR first because LDR occur already, then only LIR can occur. So the sequence is fixed already. It's like you eat already, then only shit, not shit already, then eat. Okay, so it's the same thing here. So you can start your essay like this. You can say LDR occurs in the granum. So you state where does it occur, got 1 mark. Then, as we mentioned, chlorophyll is the one that absorbs the sunlight. Now, sunlight, light energy, there's a lot of energy in it. So when the chlorophyll absorbs it, right, it will sort of like stimulate it. What we use the word here, excite it. That means it stimulates, same thing as excite it to release two things. So it's like a lot of energy injected into this chlorophyll to cause it to release two things, which are electron. Okay, so let me show you this one first. Electron, and in bio, we always mention the energy in the form of ATP. Okay, so since light has a lot of energy, it's excite the chlorophyll, means it stimulates the chlorophyll to release these two things, electron, ATP, which we will mention what does it do later. All right, now then, photolysis of water. Okay, let's start with this one here first. So photo means sunlight lah. You should know this very well already. So in chemistry bio, we always come across this word, which for my students, I mentioned this quite many times already. Lysis means what? Lysis means break down. Yes, you break down. So what I'm doing here, basically, I'm using sunlight to break down water. It's simple as that. Such an easy process. So the equation of this process is as simple as the name. There's water. There's an arrow. There's sunlight here, indicating you use light to break down the water into what? Into its ions. So one of the most basic things you learn in chemistry, what's the ions that form water? Come on, guys. What's the ions that form water? Chemia. You have what and what? H plus and also Rainier, where got O2? Ion this one last time already asked you before. Yeah, no O2 OH minus a lot of you will make this mistake, always say O2 minus, no OH minus, right? So it's OH plus OH minus 10. So that is photolysis of water. So now what we can do here is you just need to translate this equation into words. That's how you explain it. In bio, you do have the right equation, but equation will make you understand the process. Remember the process also easier. So how do you explain this? You just have to say photolysis of water occurs whereby, yeah, you mentioned the process, you got 1 extra mark, occurs whereby light photolysis water into what? Hydrogen ions, hydroxide ion. Remember, you write explanation and don't say H plus OH minus, you write the word hydrogen ions, not hydrogen, and hydroxide ions. So a bit different in your chemistry. And then in chemistry, you all learn hydroxyl, right? But yeah, here it's hydroxide. Done. Then the hydrogen and OH minus, right, each will undergo their own process. Okay, let's put it like this first. So H plus will do something, OH minus will do something. So H plus will combine with one thing to form another. So you'll write down first, it will combine with something called NADP plus to become something called NADPH. Now, we don't have to write the long name of these two substances. You can just write the short name. But in your textbook, they got mentioned what are these two. NADP plus stands for nicotinamide adenine diphosphate, which, what is it? It is actually what we call as a hydrogen carrier. Means its job is to carry hydrogen. Means its job is like the grab driver for H+. It will carry the hydrogen. So it will combine to form something called NADPH. And where does it carry it? It basically carries it from the granum to the stroma. Means it carries it for the LIR later. Because you can see LIR require the hydrogen. So this NADP plus helps the hydrogen to travel from the granum to the stroma. Like that. So yeah, the hydrogen cannot travel itself. So you need this thing to carry it. So how do we write this? Very simple also. Just have to say NADP plus combines with H plus, which is hydrogen ion, to form NADPH, which is a reducing agent. So yeah, what does hydrogen do? Hydrogen later is going to act as a reducing agent because it is going to carry out reduction later. So that you learn in chemistry, reduction requires a reducing agent. Oxidation requires an oxidizing agent. Same thing. Okay, so my H, my hydrogen, is that so-called reducing agent. Okay, so done. Then our OH. So OH minus, very simple actually. So OH minus, hydrogen hydroxide ion will become hydroxide. It's like saying chloride ion become chlorine atom. So in chemistry, you all learn, how does this happen? How does an anion become an atom? What does it need to do? Again, chemistry. Very simple thing in chemistry. Like one of the first things you learn in Form 4. It needs to donate, release or receive electron. This one you also always confuse on release or receive. You need to release, donate, lose. Yeah, so remember, chlorine is group 17. Right, chlorine atom. Ion is always stable one. So how from stable go back to atom, you need to release electron. So for the equation, will be something like this. But I say in bio, we don't need to write equation. Just use it to help you remember. So once I got OH, hydroxide, this hydroxide will combine to form water and oxygen. Now, a lot of you will wonder, how this single OH become water and oxygen? Now, actually, there are 24 OH total. Give you 12 oxygen, 12 water, 6 oxygen. Yeah, that's how we balance the thing. Then again, you all wonder, then where does 24 come from? Because initially, for every single photolysis of water reaction, I have 24 water involved. Hence, I have 24 H plus, 24 OH. That's where the 24 comes from. All right. But again, this is bio. Balancing is not important. I just let you all know only where it comes from. So how do I explain these two? Also very easy. Just translate the equation into words. So just like you write in chemistry, the hydroxide ion, how do you become hydroxide? You release electron. Since there are 24 of them, we will say hydroxide group. And for number 6, this hydroxide group, what they do, they just combine, become water and oxygen. Finish. That is your LDR, light dependent reaction. So at the end of this reaction, water and oxygen is actually your product. Because these two is basically being released out of the leaf. So I release it out. Now then, remember, this is not considered a product, even though it's something that I produce, because this thing is not released out. It's going to be used later. It's actually still inside the chloroplast, right? I just send it to the other part of the chloroplast, but not out of the leaf. So this is not considered a product. This is because I will release it into the atmosphere. Okay, this goes into the atmosphere. Done already, my LDR. So LDR basically is photolysis of water. Use sunlight to break down water into its ions, hydrogen, hydroxide ions. The hydrogen combined with its grab to become NADPH. The hydroxide ions become hydroxide groups, eventually combining to become water, oxygen. Then finally, we go to LIR, which occurs after LDR, because LIR requires the NADPH from LDR. So the first process here will seem a bit weird. Why suddenly got geometry, got maths included? Now, this is actually not maths, it's just the structure of the molecule. This is how it looks like. So carbon dioxide will combine with something called, you can write down here, called 5-carbon organic compound. There are 5 carbon, hence the pentagon shape, to form 6-carbon organic compound. Now remember, in LIR, it's mainly where I produce my glucose, the food. Glucose, the chemical formula, I believe most of you already know, C6H12O6. You need to have 6 carbon. Now in plants, they can produce these 5 carbon on their own, using proteins from the soil and so on. But they can't make it into 6. In order to make it into 6, they need carbon dioxide. That's where the final 6C come from. So 1, 2, 3, 4, 5, plus 1, 6. That's where they get the number 6 here. And since carbon dioxide comes along with oxygen, that's where they get the oxygen here. Of course, they need to balance up the equation, but we're not going to do it here. Then what about the H? Where did H come from? So the H comes from the NADPH law. So NADPH will combine with just now the 6 carbon organic compound, along with energy to produce glucose. Finish, end of story. So in my notes here, this part I left it blank. So this part, how to explain, is as simple as the equation. What plus what equals what? 1 plus 1 equals 2. CO2 plus 5 organic equals to 5 carbon organic to 6 carbon organic. So just say carbon dioxide combines with 5 carbon organic compound to produce 6 carbon organic compound. See, not very hard, right? And then for the next sentence, you cannot just say the 6 organic compound combined with NADPH, because NADPH, as I mentioned, is a reducing agent. So this is actually a reduction process. So we're going to say that this ATP and NADPH from the LDR will reduce the 6 carbon organic compound into glucose. So yeah, you need to say this, reduce the 6 carbon. Now you just add here 6 carbon. And then, just like we mentioned, daytime that one was photolysis of water. This one we need to say it is co-reduction of CO2. And that's it, done. So LIR is very short only. It's a bit complex, but very short. So if you're not confident with the answer, you can add one more here. So most plants, not most plants, but usually plants, they create glucose in excess. So they will then need to store it. So how they will store excess glucose, they will convert it to starch and either store it in the chloroplast or other parts of the plant. And that is to wrap up the whole photosynthesis process. Okay, any questions? Anything that you don't understand? Okay, right, this one. So it might seem quite complicated at first, but honestly, not at all, right? After when you listen to me explain already. So now, one more thing we need to go through is how do we obtain this equation? So this equation actually is derived from these two processes. So how do I get the equation? Number one, you need to list down the materials used in these two processes and the products. So let's list down the materials first. Means what do I use? So in LDR, what do I use? What's mainly used here? Apa yang saya guna? Number one, what do you need? You need, of course, water, right? It's called photolysis of water, right? And you also need photo, right? You need light. Now, like I said, light is not really a material, but you still include it in the equation. Yeah, so never mind, we just write it here, sunlight. Then we move to LIR. What is the main material used here? The material, I already say reduction of CO2, right? You use CO2. Okay, hydrogen is not included, because hydrogen forms from the water. Yeah, so all these are not materials, because all derives from water. Because of water, you have all this. So all this is actually come from water, so it's not included. It's not considered as a material. Then after that, the products. So what is the products here? What do you produce? Okay, LDR first. What do you produce? I already wrote for you already, right? You produce this too. So it's very, very straightforward. Water, oxygen. And what I produce at LIR, there's only one thing, glucose. So actually you can see, right, blue color here, I purposely write blue because that's the product. Red color actually is the material. And that's how you get the equation just now here. If you never wondered how you get the equation before, this is how we actually get it. And that's how we get the water here, because this water is produced at the end of LDR. Like I say, it produces lesser than it requires. That's how we get the equation. But you will not require to balance this equation because it's not chemistry, it's just high school bio. But you must know how to write this equation, because kind of question will ask you to write the word equation. In bio, usually we are required to write word equation rather than chemical equation. So instead of writing H2O, you write water. Instead of CO2, you write carbon dioxide, of course. Oh, and one more thing you need to include here, chlorophyll. Because sunlight is absorbed by the chlorophyll. Sunlight on top, chlorophyll bottom. Because sunlight is in the sky, chlorophyll is on the ground. So that's why it's written like that. Legit one. Okay, so once we're done with this, this is one of the most common things they will ask. I mean, most basic thing you will ask, explain the reaction. Later, we will cover slightly harder punya question, but you also require knowledge from these two reactions. So in a while, we'll do that. So let's finish up our notes here, page 16. So now that we know LDR, LIR already, you must also know how to compare them. Again, can come up for both assay and structure. If assay can come up to 7 to 10 marks. Yep, so number one, light energy, we already know, of course, LDR requires it. LIR does not. Time of reaction means what time of the day it occurs. Light, of course, only can occur during the day. But even though light independent does not require light, it also occurs during the day and also during night, but only early parts of the night. Because after, let's say, around 8, 9 p.m. like that, what happened here is, because nighttime, you don't have this reaction. Means even though LIR can occur without sunlight, the NADPH will get lesser and lesser. So maybe around 1 or 2 hours later, the NADPH finish up using already, then LIR also will stop. So even though I say it can occur during night, but only small portion of the nighttime. Site of reaction, this one is the first thing we mentioned already, Granum stroma. Type of reaction is the photolysis reduction era. So this is photolysis of water, reduction of carbon dioxide. Substance required, basically you take the equation and then separate it like how we separate just now. So substance required is basically water. So substance here is not included sunlight. Sunlight is not a substance. And here's CO2. Product, what do I produce? Water and oxygen. Product here, glucose. Photolysis of water, okay, this one nothing much, occur, does not occur. ATP, hydrogen, you can say this one uses ATP and hydrogen. This one produces. The other way around, I'm so sorry. Use, produce. Yeah, LDR produces this too. LIR uses this too. Can? All right, so if can, come. Next question, next part. So just like transpiration, the surrounding factor also can affect how fast photosynthesis can go. However, for photosynthesis, right, factors is not the main thing, because unlike transpiration, right, photosynthesis has a process. Yeah, so question will focus more on this part actually, rather than this. But yeah, you usually need to know because there will be still question asking. So for photosynthesis, there are only three factors: light, carbon dioxide, temperature. Humidity does not really affect it, along with also air movement. So light intensity, of course, more light, more photosynthesis. So if question asks you to explain like how light affect photosynthesis. So of course, yes, I know more light, more photosynthesis. Why? Because when there's more light, means more LDR occurs. More LDR, what process in LDR needs light is the one that we mentioned just now. So just continue. More photo is this of water occurs. Easy. Now, however, if you look at this graph, beyond a certain point, which they indicate here, point R, beyond that, right, if I still continue to increase light, your photosynthesis remains constant. Why? So as they mentioned here, does not increase in light intensity, does not increase the rate of photosynthesis, because in plant, they have limited amount of chloroplast. Means there's limited amount of sunlight that they can absorb. It's just like my bottle have a limited volume of 1 liter. If I pour 2 liters of water, I'm only going to take the first liter, but the second liter of water that I continue pour will not be filled by my water bottle. So if let's say I can absorb this amount of sunlight only because I have this amount of chloroplast, if the sun now very hot, got this amount of sunlight, the remaining extra here cannot be absorbed. All right. So at point R is the so-called saturation point. Means the maximum point, maximum amount of sunlight that it can absorb. So at this point, we will say the other two factors, which are temperature and carbon dioxide, becomes the limiting factor. Means in order to increase photosynthesis, I need to increase other factors already, temperature, CO2. So maybe at this point, temperature and carbon dioxide is still not maximum yet. So this two is the one that limiting, slowing down my photosynthesis. So this two needs to increase, then only my photosynthesis can further increase. Okay, so if they ask you to explain this part, we use this point to explain these 2 points here. All right, then for CO2, pretty much the same like light intensity. There's also a fixed amount of carbon dioxide that can absorb. So hence the graph like that, same shape. So for the explanation, the balik law. So just now light is required for LDR, but we know carbon dioxide is LIR, right? So we'll say more LIR occurs. More LIR means more LIR punya process law, reduction of CO2 occurs. And same, this point, same explanation. Increase in CO2 after 0.9 does not increase the rate of photosynthesis because same thing, I reach saturation point, maximum point, and then we say the other two factors becomes limiting factor. And then last, temperature. Now, don't copy first, listen to me first. So if you look at temperature, the graph is slightly different than the first two because this one, yeah, at first I increase, but up to the maximum point already, after that, I won't remain constant. But if I further increase, it would decrease the reaction, the photosynthesis. The reason is because photosynthesis, which is LIR specifically, the independent reaction, light independent reaction, requires certain type of enzyme, which we just call it as photosynthetic enzyme. So if you remember your form 4, this is how enzyme reacts to temperature. From low temperature to high temperature, with an increase of temperature, yes, my enzyme reaction will increase. But up to a certain point, what we call as optimum temperature means the most suitable temperature. After that, if I further increase, the reaction will decrease. Okay, because beyond optimum temperature, we already know your enzyme basically will be broken down. High temperature, your enzyme cannot retain its shape, it will just break down. So for this part, how to explain? You just say that this higher temperature, higher rate of photosynthesis, because every 10 degrees increase in temperature would double the rate of photosynthesis until optimum temperature, right? Which is, for plants, usually is 25 to 30, depending on the type of plant. Some can even be more. So beyond optimum temperature, this part here, oops, here's the explanation. Remember your form 4 or not? Temperature too high, what happened to your enzyme? You cannot say enzyme breakdown, enzyme spoil, enzyme die. What was the term you need to use? Yeah, you all still remember, very good. You must use the word denatures. It's a term used to describe your enzyme being broken down by excessive heat. And therefore, your light LIR stop, no LIR, no glucose. Of course, photosynthesis cannot be completed. Right? Yeah, so write this down, and then we'll proceed to the next page to do, as I label here, higher level punya photosynthesis question. Okay, yeah, but you write this first. Right, so done. Yeah, so for the questions at page 17, these are slightly challenging punya photosynthesis question, but also do expect it to come up. Yeah, so we will move to page 17 once you're done. Done already, guys? Can Yeah, so behind the question, actually, we won't have time to do lah. So what we write down here actually is basically quite a lot of it already. So yeah, the other question you try to do yourself. Okay, let's start with this one. So this one we did, for my students, we done this before already. See you all still remember or not? So they say if a plant is exposed to sunlight for 24 hours a day, explain how does this affect the light independent reaction of the plant. So 24 hours of light, how does it affect your LIR? So LIR, remember, is independent from light. So do you think it will increase my LIR or decrease? What do you think? Remember or not, we mentioned just now, previously. Will it increase or decrease? So at first glance, a lot of you say, oh, if a lot of light, then LIR, I don't need light, so it will negatively impact. Some of you will think like that, but actually no. The answer is it will benefit it. It will actually increase. Okay, like some of you mentioned. So those are the decrease. Okay, never mind. So because remember, LIR depends on LDR. So if let's say LDR stops, there will be no NADPH. No NADPH, my LIR cannot complete, right? So if there's 24 hours of sunlight, this 24 hours supplies of NADPH means 24 hours, my LIR can keep on occurring. So actually it's good. So what you need to say here is your rate of LIR increases. And then you explain why, because when there's more sunlight means more, you can say more sunlight for LDR to occur. And what is the main process in LDR that needs sunlight? Photolysis. So we just continue to explain. I'm going to write more than 3 points here. So more photolysis of water occur. So more photolysis of water means more what produced? The two ions, the OH and H+. Which question is this? It's the same after page 16. Page 16, page 17. There's on the notes. It's in the notes, not in the question. The same thing just now. The same book as notes. Okay, Wesley can. So more photolysis of water. So therefore more H+ and OH ions producer. So how does this link to LIR? LIR needs the OH. Needs the H, the NADPH. So from here, then I will link to my LIR already. So I don't say more oxygen, more water, because that does not link to LIR. To link to LIR is this further. So more H means more NADPH is produced for reduction CO2. Right? Now for the next one, now this will be quite challenging, but it's actually inside your textbook one, which is how different color spectrum of light means how different color of light affects photosynthesis. So now actually it's not that you simply shine any random light on the plant, then it can carry out photosynthesis. So you can see sunlight actually has 7 colors inside it, right? If you study physics, you know there's the rainbow color, basically red, orange, yellow, green, blue, violet. So in sunlight there's just 7 colors, but out of these 7 colors, the both color at the beginning and at the end of the light spectrum is the most absorbed color light by the chlorophyll. So we know chlorophyll is actually green. So since chlorophyll is green, right, it will reflect color spectrum that have similar wavelength towards it. Yeah, I think in physics also you'll learn this. Different color have different what we call as wavelength. That's why our eyes interpret them differently. So green is right in the middle of the color spectrum. So basically, colors that have similar wavelength towards green, which is green itself, blue, yellow, like that, will be usually reflected. But color spectrum have larger difference wavelength, like you can see, purple and red, are absorbed by them. So sunlight, even though you cannot see these 7 colors, but when sunlight is reflected, sometimes you can see the 7 rainbow colors come out. Yeah, that's why at the end, after rain already, when the sunlight shines on the water on the ground, it reflects as the rainbow color. Yeah, that's basically it. So in this experiment, you can see, I put a plant under water. which I want to test photosynthesis, of course, this experiment. And I shine these two colors here. See, violet, which is purple, causes 50 air bubbles to be released. Green color causes 10. So first things first, what gas is this air bubble being released by the plant? So we are studying photosynthesis now. So what gas do they release in photosynthesis? Yes, exactly. Georgina, Nandini, Shinpei, very good. It is oxygen. Yes, Rainy, correct. Also, oxygen. Okay, because photosynthesis release oxygen. So when got light, it carry out photosynthesis. So the gas produced here, same is oxygen. So more bubbles means more oxygen, means more photosynthesis. Common sense. And this one, the opposite. So for this question, they ask, based on table 2, describe the effect of different colors of light in the light-dependent reaction of photosynthesis that occur in the plant. Sorry, the word there, in the plant in beaker R and in plant in beaker S. So they want you to focus on LDR, light-dependent reaction, because oxygen gas is produced during LDR, right? So we only need to focus on that part. So you can start your answer like this. So let's talk about green color first. So you need to separate answer green and violet. So for green, you can say like this. Chlorophyll is green, which reflects light spectrum that have similar or small difference in wavelength. Okay, so therefore, the chlorophyll will absorb lesser light energy from green light. So it reflects light spectrum that have similar or small difference in wavelength. Okay, or if you really can't remember this, you just say chlorophyll is green, which reflects green light. So this will be slightly more technical punya term, because green light has small difference in wavelength, or the same. So this way they don't absorb it, they reflect it. So if I reflect means I don't absorb light energy. So no light energy means what? No LIR, no LIR, no photolysis, then just continue until you link to oxygen. So how do I do that? Okay, you just say, therefore, lesser LDR occurs. No LDR means no photolysis, right? So we just continue, lesser photolysis of water occurs, and therefore lesser the two ions of OH minus and H plus produced. And then how do I link to the bubbles? So as we know, OH is used to form the oxygen. So then we can say lesser oxygen gas release. Or actually, if you start from here, right, it's already good enough already. Yeah, I'll just give you one additional point, the first one. And then for the purple one, is the opposite. Since purple have larger difference in wavelength, as you can see here, they are quite far apart, it will absorb it. Okay, so since they have larger difference in wavelength, so therefore, the chlorophyll absorbs more, so it was less mass, so now more light energy from violet light. So the same thing, if you absorb more light, means now the entire opposite. You have more LDR, you have more photolysis, we have more of those two ions produced, and more oxygen. So just the balik only. So you can see all my points here is just LDR only, no LIR involved, because oxygen gas is in LDR, produced in LDR. Okay, can? Yep, and that's how we do this one. So both of these questions, it's not like explain LDR, LIR type of question, but you can see you need to use the knowledge, the processes that we learn in LDR, LIR to actually answer them. Okay, can I? All right, so almost done already. We just go through a bit of compensation point, then I'll show you like 1 or 2 questions and then we're done already. So the last thing in this chapter is called compensation point. It is actually the easiest thing, but to understand compensation point, first you need to understand this graph. So by now, if you don't know how to look at this graph, okay, let me just show you real quick. It's very simple. Look at the x-axis, light intensity. Oh yeah, by the way, what is compensation point? Compensation point is actually to show the relationship between photosynthesis and respiration in plants. Yeah, plants also undergo respiration. All living things, need to undergo respiration. So these two reactions are the complete opposite. Photosynthesis uses carbon dioxide to mainly produce oxygen and glucose. But respiration, as we know, uses oxygen to oxidize glucose to produce CO2. So this compensation point, this graph, right, actually is trying to show you these two. So for the x-axis is light intensity. So of course, if we move from the left to the right, light intensity increases. So at the right side, light intensity is the highest. It's like probably like 1 p.m. or 12 p.m. Then here the light intensity is lowest, of course, during nighttime. So if you look at the y-axis, there's a positive side and negative side. So for the positive side, carbon dioxide, they say here uptick, increase. Uptick means what? Means I absorb CO2. Here is the carbon dioxide release increase, so I throw out carbon dioxide. So it's the opposite of intaking, so that's the negative side. So if I ask you one thing, between photosynthesis and respiration, so if you look at this part of the graph, this line here, is this photosynthesis or respiration? Okay, come on, I'm going to finish already. I know you're very tired. Is this photosynthesis or respiration? Come on, almost done already. This part, you can see, is positive side. I'm absorbing carbon dioxide. Absorb carbon dioxide is actually photosynthesis. So this whole part here is photosynthesis. All right. But if you look at this part here, this part, the line here is indicating the plant releasing carbon dioxide. So release carbon dioxide is actually respiration. You can look at it like this, or you can look at the light intensity. So just now I show you, straight away look at the carbon dioxide, or you can switch the other way around. You can look at the x-axis. So you can see here light intensity is low. Light intensity low, of course, is respiration, right? How can light intensity low? Photosynthesis. So as I increase my light, you can see my graph, right, slowly going to move towards the photosynthesis part. Because this is like, for example, let's say the sun from 5 a.m., 6 a.m. to 7 a.m., the sun is rising. So I start to more and more photosynthesis, right? So when let's say I reach around 7 a.m. like that, when respiration decreases, photosynthesis slowly increase. So around 7 a.m. like that, when the sun is not too bright, there'll be a point whereby photosynthesis and respiration is the same. So that is what we call as compensation point. Okay, so before compensation point basically is respiration, and after is mainly photosynthesis. So you can see here, before compensation point, since light intensity is low, so rate of respiration is higher. If rate of respiration is higher, the rate of carbon dioxide release, of course, is higher. You release more carbon dioxide during respiration. And respiration needs oxygen, so you absorb more oxygen compared to releasing it, and you use more sugar. So as I say, as the light intensity slowly increase, photosynthesis also increase, respiration decrease. So when they are same at time, that is called compensation point. So this is where question always like to ask. They ask you to explain compensation point. So how do we explain? All this point, same only. So now your rate of respiration is the same as rate of photosynthesis. And everything else is also the same. Carbon dioxide release, same as carbon dioxide absorbed. Oxygen release, same as absorbed. Sugar also the same. Okay, so as I slowly move towards later of the day, 8 a.m., 9 a.m., 10 a.m., when you get really hot already, sun really come out already, then the plant will focus more on photosynthesis. As you can see, photosynthesis increase as the light intensity increase, which makes sense, right? But even though photosynthesis is at maximum, right, there is still a bit of respiration because you still need to produce energy to survive. So it's not necessarily at here respiration is 0. You still got a very low compared to photosynthesis. So basically here is here. So everything terbalik now. Photosynthesis higher. You absorb more carbon dioxide. You release more oxygen and produce more glucose. EXP means explain. So they ask you to explain compensation point. Yeah. So this is how we answer. So here I believe you can fill in by yourself. Okay. So actually what I want to teach is just under here, but don't leave yet. Let me just go through one question real quick. Okay, we go through a bit of one photosynthesis question, page 21, this one here. So I want you to understand a bit this diagram. So photosynthesis, sometimes they will use this diagram. So a lot of photosynthesis question, right? They will use like put LDR, LR inside in the how to say in the flow chart form and ask question. Which the chart here is actually has the same equations that I wrote for you just now in front of. So they say based on diagrams, they state 2 raw materials needed in photosynthesis. So actually for this question, you don't even need to look at the diagram because you know the 2 raw materials you need for photosynthesis is what? The equation just now. You need what and what, guys? Come on, go to finish already. Wait. You need water. That's for the LIR one. And you also need carbon dioxide. Easy. Then states reaction X and Y. So try to look at the diagram and then try to identify X is what, Y is what. So you can see X got sunlight. So by looking at this, then actually you don't need to look behind already, which of course LDR, right? LIR law. LDR, LIR. So if you look at the equation here, see the same thing. See sunlight break down water into 2 ions. The hydroxide ion become water and oxygen. Then the hydrogen become NADPH and use in your, what's this, LIR law. Exactly the same. If you wrote photolysis of water, cannot, because it's a reaction, so it's either light-dependent reaction or light-independent reaction. Unless they ask you to state the process that occurred in X, then only can say photolysis. Okay, right. So one product in reaction X and Y. So this one also very straightforward. Product, oxygen in X, Y, glucose. Okay, we finished these 2, then done already. D and E. Name the process. All right, so can you guys tell me what this do? So the first one, water becomes hydrogen hydroxide actually. It is, today we learn what's the process of water being broken down in these 2? No, you can't say LDR because they're very specific, right? LDR is the whole thing. This is only one part of LDR. Yeah, so correct. Photolysis, not hydrolysis, photolysis. Okay, can. So the next one, glucose become starch. No, glucose becomes starch is not reduction. Wesley, correct already. So just look back at your notes, got mentioned one. And by the way, this is in form 4 reason 1. Glucose becomes starch. Smaller molecule become larger molecule. This is actually, yes, condensation. Remember, starch broken down to glucose is called hydrolysis. This is condensation. Form 4, chapter 4. Last one, explain how NADPH is formed and explain its role in photosynthesis. How NADPH is formed? Okay, so we know, right? NADPH is formed from what and what? NADPH mainly has H in it and NADP plus in it. But here I write electron because electron is used to form the NADP plus. Yeah, so very simple. You can say NADPH. You can say hydrogen ions combine with NADPH to form NADP plus, sorry, to form NADP. And its role in photosynthesis. So NADPH is a what? You must say what it is and what does it do. Yeah, it is a reducing agent. So NADPH is a reducing agent. And who does it reduce? It reduces. So back to our notices now, or actually you can just look at here. It reduces this into this, which is your 6th carbon, right? Along with the ATP. So you can say it reduces the 6th carbon organic compound. You can say along with ATP to produce. Actually, no water here, lah. Glucose. Only. All right, so that's it for today. Other questions, please go and try to do yourself. Answers are already, when you receive those answers will be together with it. If not, again, you can click the link that I posted just now, which I will do it one more time. Answers are already there, and you can do the questions yourself. Okay, so that's it for today. Hope that the revision actually helps you understand chapter 2 more. So if you are my student TGC 1, we will read, these are the things you're going to revise in May, okay? Form 5 chapter 1 and also Form 4 chapter 14. But if you're not my student, if got chance, hopefully we will see each other again. And yeah, good luck. See you. Bye bye. Bye guys. Rest well. Sleep. Good night. Bye bye.